This is section 17.5, entitled The Genetic Code and Protein Synthesis. We've talked about nucleic acids, we've talked about DNA replication, we've talked a little bit about RNA transcription and the transcription of genetic material, the genetic information in the DNA transcribed to messenger RNA. Now we're going to talk about the protein synthesis, which pretty much finishes off this basic description of protein synthesis. Actually, the nucleic acids do a few other things than that, but this is what we really this is what you really need to know here. The genetic code is a collection of nucleotide triplets or triads or something like that, sequences of 3 of these, each of which codes for a protein, and actually a couple of them code for a stop codon or a start codon. And the translation process we just started here, where what they have here, they have a transfer RNA for serine, a serine transfer RNA, which transfers a molecule of serine, the amino acid serine, to the growing protein chain. And this particular one has an anticodon, an anticodon as they put it, over in that little red, the 3 nucleotides marked in red. So those are, it's AGU, and remember we're in RNA now, so we don't have thymine anymore. We have uracil. And the complementary bases to the anticodon are the codon. Actually, more properly, we probably should call the anticodon complementary to the codon, because the codon is what brings the information in. And you see the codon is UCA. That codes for serine. So you have an activated transfer RNA, which essentially means that it has its amino acid attached to it, and it attaches to the messenger RNA at an anticodon with AGU, the sequence, and UCA, of course, is the sequence on the messenger RNA codon. And that's part of the process of synthesizing the growing protein chain. Now, the genetic code itself, as I said, it's a series of 3 nucleotides or triplets in messenger RNA that are called codons, and each one specifies an amino acid. Now, in your text, and this should be also in here in the slide too. Let me just a moment. I know it's here. Yeah, there it is. Okay, it's there. You're going to see that in a couple of slides. This particular sequence UUU that they give is really the first sequence that was definitively found to code for an amino acid. Phenylalanine was the first one that was identified, because it just happens to be the one that is translated from the codon UUU. Now, this is actually a rather interesting situation, because I put a thing on the discussion board on this. You're going to see that I did mention a start codon, okay? And well, I'll pick that up when I go to the next slide after this one. The codons have been determined for all 20 amino acids, and the AUG signals the start of protein synthesis. Okay, it's that when the RNA polymerase is in the, or actually when in the enzymes are making the protein, and this is all going on in the ribosome, the ribosome reads AUG at some point, and it's gonna tell it to go. Okay, and we start here. AUG also codes for methionine. So the first amino acid in every protein is essentially methionine. Most of the time, it might be removed enzymatically, but it's going to start the initial protein that comes off the ribosome is going to start with methionine. Eventually, like I said, it might be removed. Proteins do get modified after they're synthesized. All right. So if you think about the number of codons possible, you have 3 letters in each word, if you want to call it that, each codon, and you have 4 choices of a letter. You have adenine, you have guanine, you have cytosine, and you have uracil. That's 4 each. So 4 times 4 times 4 is 64. There are 64 codons possible. So there's plenty there to code for all the amino acids. And there are, in addition to the start codon, which also codes for methionine, there are actually 3 stop codons. So when translation is going on, when the mechanism in the ribosome finds a UGA, a UAA, and a UAG, the protein synthesis stops, the protein chain comes off, and the synthesis is over. All right, we have that. Now, what I was going to say, these are all the codons and what they code for. It turns out that when this was first done, when this was first discovered, the polyuracil, they just made a polymer of uridine phosphate. Okay, that nucleotide. They just polymerized that nucleotide, and they found that when they put the enzymes in and the transfer RNAs and a mixture of amino acids, they found that what they got was a polypeptide of phenylalanine. Now, this is a little odd because you notice that there's a stop codon or start codon, and why do you need a start codon if they can just polymerize this with just a couple enzymes? Well, what happened is that apparently they just fortuitously stumbled on a set of experimental conditions that somehow relaxed the requirement for the thing for this to start at a stop codon. Of course, this wasn't on a ribosome. This was just enzymes and RNA and protein and amino acids and so forth. So it was actually a stroke of luck. Otherwise, because as it is, it took years to work out this code you see in front of you. And a couple of things to note about it. Most of the amino acids are coded for by more than one codon. Okay, you see phenylalanine, there's 2, UUU and UUC. There's, I think, a couple of them have 6. Serine looks like 4. Valine looks like 4 there. There's actually a couple have 6. I can't find them here. Yeah, okay. There's leucine has 6. You can see them in the U column. The U, it's actually, well, not the U column. The C row. Let's put it that way. The U row and the C row. There's 2. The UUA, UUG code for leucine. The CU, all the CUs, CUU, CUC, CUA, and CUG code for leucine, so that's 6. And if you look at the second column at the top, where UCU, UCC, UCA, and UCG code for serine, and if you look over in the 4th column in the A row, the 3rd row down, you'll see there's 2 more codons for serine. So it varies quite a lot. There's a couple of amino acids are coded for by a single codon, and there's, for example, methionine is coded for only by ACG, and it looks like tryptophan is a single codon, UGG. It's So that it varies how many codons each amino acid will have, and it usually it tends to be the more common amino acids are going to have more codons, but there is quite a bit of redundancy in this genetic code. These are the sequences in messenger RNA that are derived from the DNA, the gene in the DNA, and they are used as the template for the transfer RNAs carrying the activated transfer RNAs carrying amino acids. And we'd like you to be able to determine amino acids from the codons, and it's really pretty simple. Given a sequence of nucleotides in a messenger RNA, and they will tell you if it's a codon or an anticodon, and you'll see. I'll go into this a little bit deeper. You see the codons are in messenger RNA are given here as CCU, AGC, GGA, CUU, and if you're asked to identify the amino acids, you're simply going to take each three, the first three, CCU here, and look it up in the genetic code. What does CCU stand for? Okay, it's proline. AGC, you look at the first letter and is A, so you look in the A row, and then AGC, so you've got G, so you look in the G column, so and then find AGC, and that's serine, okay? So that's going to be serine. GGA is glycine, CUU is leucine, so you're going to have those four. Now I'm going to just do a little whiteboard thing here just to clarify some of this. Okay, here's what we had. There was a problem we started with, and something you might actually be asked to do something like this, to go from these codons. These are the messenger RNA codons. This is the genetic code that you see reproduced in the previous slide and also in the table and in the text. It's page 597. It's in section 17.5. It's from that table. Okay. This comes from the DNA, and this is synthesized in the nucleus from DNA as a template and in the process called transcription. That's going to be the first step. The second step is going to be translation. You see what's happening is the DNA, you're essentially copying the sequence of the DNA to another set of nucleotides, to a nucleic acid, and then you're translating it into a protein at the end of the process via the transfer RNA carrying the amino acids in. And this information is going to be useful in the next few pages as well. Okay, so the transcription process, you're translating DNA and the codons, or let's put this thing in here, these lines in here. These codons are going to be translated or transcribed from DNA. Now, you're going to have, remember, CG is these are going to be from complementary base pairs. What's going to happen is these amino acids or the ribonucleotides are going to line up with their complementary base on the DNA, and in this case, this is going to be this DNA is going to be G, and U is going to be complementary to A, and the AGC. Okay, now it's going to be T instead of U because this is DNA. Remember, and then C and G, and then it's going to be C. A is going to be, it would be uracilin RNA, and remember, it's thymine in DNA. Okay, this is going to be the DNA sequence, and then we have G, and then U is going to pair with A. Okay, so that's going to be the sequence in the gene. And then when you translate it, these are going to be anticodons now. So this is going to be GGA, and we're going to have, now it's going to be UCG. I get this right. I think I got that right. Yeah. CCU, GAA. Okay, those are going to be the anticodons. And these are going to translate, as we saw, these translate into proline, the sequence proline, proline, serine, glycine, and leucine. This is a lot to keep track of, and I'm putting this up here just to let you know that it is a lot to keep track of, and the codons, what's in the genetic code, are going to be the codons in the messenger RNA. So then there's going to be what's in the gene is going to be transcribed to that, and then the transfer RNA is going to carry the amino acids in, and they're going to be turned into proteins. Okay. All right, so we go on here. Anyway, we saw, wait a minute, write the order of amino acids coded for by a section of messenger RNA with this base sequence. Well, they say it's messenger RNA, so these are codons. Okay, so this is the genetic code. All right, and they're giving you a number of choices here, and you have to pair the codons with the proper amino acid. Yeah, so GCC corresponds to alanine, GUA corresponds to valine, and GAC corresponds to aspartic acid. So you're going to write this as a sequence of 3 amino acids, and you already know how to do that. You know how to write a tripeptide sequence, and so there's your tripeptide sequence. All right, alanine, valine, aspartic acid. Did I say aspartic acid? I think I did. But GAC, it is aspartic acid you see there. Okay. Now, in order to get this to happen, you have to activate the transfer RNA. And what that essentially means is that an enzyme, amino acyl transfer RNA synthetase, there is a specific one for each transfer RNA. So there's going to be amino acyl transfer RNA synthetase. You're going to have serine transfer RNA synthetase. You're going to have a tryptophanyl transfer RNA synthetase, and so forth. So every amino acid has its own transfer RNA, and that transfer RNA has its own amino acyl transfer RNA synthetase. So you've really got a lot of things going around in the vicinity of the ribosome, with all these transfer RNAs and amino acids and a lot of reactions going on. Okay, so what you get is an ester bond at the acceptor stem of the transfer RNA, and there's a couple of diagrams around here. So that lands on the ribosome, or that transfer RNA lands on the messenger RNA, and they line up, and there's enzymes which are going to detach the amino acid from the transfer RNA and attach them to the growing protein chain. And then after that's done, the transfer RNA goes off and either, I don't know if it's reused or just broken down, and the nucleotides reused. You have your initiation step, of course, where the start codon is going to form these, when they say hydrogen bonds, that's that base pairing. Okay, the AUG is going to form the hydrogen bonds to the anticodon of the methionine transfer RNA. Okay, that's going to be the first step. and then you're going to have another transfer RNA is going to bind to the next codon, placing a second amino acid there, and then a peptide bond is going to be formed between those two. So you're just going to keep forming peptide bonds as you go along. And again, here's another picture of that transfer RNA. Okay, so we've already talked about the first step in the protein synthesis, where you have the start codon and then a second amino acid comes in, and this is just a picture of an activated serine transfer RNA. As you can see, we've talked about that already. Okay, so once the first bond is formed, I did mention in an earlier video that the ribosome is like a tape reader, and it moves along the messenger RNA, and it helps it is instrumental in assembling the protein from the appropriate amino acids in the appropriate sequence. This is called translation, as the initial transfer RNA detaches and then moves to and then the ribosome moves to the next codon, and the next transfer RNA attaches, and it moves codon to codon, so the transfer RNAs can keep attaching new amino acids. And of course, some proteins are pretty large. Some have, I think, tens of thousands, thousands of amino acid residues. So it's going to go on until it reaches stop codon. Anyway, this is sort of I'm going to go to the chain termination first. The termination step is you have all the amino acids are linked, and the ribosome reaches a stop codon, which is UGA, UAA, or UAG, and there's no transfer RNA for the stop codons. So what simply happens is that the polypeptide detaches from the ribosome, and it's like it says here, usually methionine is removed from the beginning of the protein chain, because it's in most cases it's there only as just to indicate the start codon, but it is there at the outset. Okay, what we had here is really a diagram of the entire process. So let's blow this up a bit. I can make it bigger than that. See if 200 is too big, 200 is too big. Let me make it around 150. Let's try 150. And it's still pretty large, but I didn't want to do that either. Just bear with me a moment here. Okay, I didn't want to move it, so let me shrink this down to a little more. That's at 150. Let's try 125. See how that works. Yeah, that looks a little better. It's sort of readable here. Okay, so here we have a summary of the process. This is in your text. There's 5 numbered steps here. 5. And it starts in the nucleus, where the messenger RNA is synthesized using DNA as a template, and you see the bases that are pairing, and you see the gene itself. You could see the bases, or the nucleotides that are the ribonucleotides, what's going to become the RNA, is pairing up with the ribonucleotides in the gene in DNA, and of course the other strand is just a template strand for the DNA. So once this is done, once the messenger RNA is synthesized and it's on its way out of the nucleus, those two chains, the DNA chains, snap back together, and the DNA is reformed, and this gene can make more transfer or messenger RNAs as need be. Okay, so the RNA is going to leave the nucleus, and it's going to attach to a ribosome, and you see it's going in. The ribosome is going to come in it's moving from left to right, the way this is written. Okay, so you have that there's a portion of the messenger RNA is already translated. Okay, so what's going to happen is the messenger RNA, after it gets out of the nucleus, it's going to attach to a ribosome, and the ribosome's going to read it, kind of like a card read, not a card reader, a tape reader. I don't know if they still use those anymore in computers, but they used to be very common. You put the tape in, and it just would just run through, and whatever was there'd be little holes punched in the tape, and they would that would code for, well, it would be your computer program or some other information. All right. So what happens, and once the messenger RNA gets attached to the ribosome, you have incoming transfer RNAs, which are they hydrogen bond to the complementary base in the appropriate codon. And remember, the transfer RNA is an anticodon, so they bind to the codon in the on the RNA, the messenger RNA, and two of them get lined up. Then an enzyme will form a peptide bond between those two adjacent amino acids, and then the ribosome will keep moving on. The transfer RNA, which is no longer activated, it no longer has a they no longer have those transfer RNAs after the synthesis, after the amino acid's been removed. They're now inactivated, so they go off into the cytoplasm, and they can be reused. And in the end of it, you're going to have this protein chain. So we can move on to the next slide, and I can fit. So in the termination step, you have, okay, all the amino acids are linked, and you reach a stop codon. Ribosome reaches a stop codon, and there is no transfer RNA for a stop codon. So at that point, the polypeptide detaches, leaves the ribosome, it breaks free of it, and goes off to where it's going to be used in some organelle. Maybe it's going to be secreted into, if this could be, it can get in, it'll go through the, it could be secreted into, I don't know, like the intestine or something like that, depending on where these cells are that are actually synthesizing this protein and what the protein is going to be used for. And the initial amino acid, which is always methionine, is usually removed. So you're not often going to see methionine as your initial amino acid in a protein, but it does happen. But you're usually removing the methionine. So it's the methionine has the same codon as the start codon. It's the methionine and start codon are the same codon, so that's ACG. And if this is in the middle of the chain, of course, it's not a start codon. The ribosome's already reading it, so this is not a signal to start. It's going to be a signal to put a methionine in. Actually, there's I guess I'm gonna mention this. Usually, these genes are linear. There's just one, or a gene, a genome, the DNA is usually going to have a sequence. A given sequence is going to be coding for one protein. There are a couple of bizarre situations, and that have been discovered, where a virus has actually overlapping genes, and in other words, the same region of the DNA is going to code for more than one protein. It's going to code for two. That's very exotic. That doesn't happen. We're not going to see this here, but it's those proteins actually contain a lot of AUG. I said ACG. I meant AUG. They're going to have a lot of these things are going to have a lot. Let's put it this way: some of these proteins are going to have a lot of methionine in them, and then they're going to have a lot of the sequence for the start codon, and if you think about it, I'm not going to go into any detail on this, but what will happen is there could be two reading frames that want to superimpose on each other. And if you think about it, though, to get a third one on is probably going to be very unlikely, because let's take a look at the sequence in the stop codons. You see AUG is a start codon, UGA is a stop codon, and if you were to shift if you had a sequence of AUGs and you shifted it over by one, you would go from a sequence of AUG to a sequence of UGAs, which would be a stop codon. So there's only a limit to how far this overlapping genes can go. And I only mention it because it's one of those curiosities that you find once in a while in biochemistry. So, but that's not a concern here. Chain termination. Well, we've mentioned all this, so move on. Termination. Once the polypeptide is released, the R group, the amino acid, and the new protein chain, the R groups, the protein's going to fold into ultimately it's going to be folded either partly spontaneously, partly there's helper proteins that'll actually help it to fold into the proper configuration, and then you'll form, you're going to form the various cross-links, salt bridge and disulfide bonds are going to have to be formed enzymatically, and the protein's going to be assembled into not only primary and secondary, but also the tertiary and quaternary structures. So if you have like hemoglobin, you're going to need 4 subunits there, so you're going to have, and there's 2 pairs of identical subunits, so you're going to have a couple of different proteins, polypeptides synthesized by a couple of different messenger RNAs, and then they're going to go on from there. Okay, so these are the steps in protein synthesis. We've gone through them in some detail. There's DNA transcription, which produces a messenger RNA from a DNA template. Then we have activation of transfer RNA, which is when the transfer RNAs, each one is specific for each one is specific for a specific amino acid. So the tRNAs pick up their amino acids and are activated. Then the messenger RNA binds to ribosome. The ribosome starts reading it, and sequentially the transfer RNAs land on the messenger RNA, and starting with the first the start codon, the methionine codon, and the successive transfer RNAs bind. They just went from one end to the other, and then it hits a stop codon, and the protein's released, and the transfer RNAs are off. They'll get recycled or used for another protein synthesis or something. Okay, and then complementary sequences. Now we have, I did this already. I did this on that whiteboard. They add here, this is a different protein, I think, than was there. But this is the whole sequence of what's going on. You have the informational strand and the template strand. All right, so the informational strand, see, a while back when I think I still have this up here. Let me take a look. Yeah, you see the DNA gene. This is the template strand. Okay, in this. So I've actually, I don't have the whole sequence in here, but here's the whole sequence. So you have the informational strand, which actually, if you look at it, it actually has the same essential strand, or the same essential sequence as the messenger RNA, except that in DNA you have thymine, and in messenger RNA you have uracil as the base in each of those nucleotides. So you see the informational strand is complementary to the template strand. The template strand is what's read by the RNA polymerase, and then it gives you the messenger RNA, which carries a template for the transfer RNA, and the messenger RNA is carrying the information, essentially, if you look at that, the way it's working. The transfer RNA has anticodons, which are complementary to the codons in the messenger RNA, and then this sequence will be converted into a polypeptide chain. Okay, now let's do some learning checks here, or a learning check here. Match each of the following terms to a definition or concept. You have to pay attention to these, by the way. I've had some issues with a few of the problems that in the homework. They're maybe not issues necessarily, but they're very specific. Okay, you really need to be accurate when you're looking at these. So, okay, match the following terms. Ribosomes moving along messenger RNA, adding amino acids to a growing peptide chain. All right, well, there's got to be translocation. There's some movement going on there, right? A completed peptide chain is released. That's going to be termination. It hits the stop codon. Synthesis is over. Protein chain comes off and goes off to where it's going to be used. Transfer RNA attaches to its specific amino acid. Remember, that's activation. Activation is when the amino acid is attached to the transfer RNA. Transfer RNA binds to the AUG codon, which is the start codon, and also methionine, and that's going to be the initiation step. So there's going to be you see a couple of these, activation and initiation, look like they could be similar. You need to keep those separate. You need to keep that in mind. They are different processes. And here we have it: translocation, termination, activation, and initiation. Another learning check. The following sequence of DNA is used to build messenger RNA for a protein. What is the corresponding messenger RNA sequence? Well, we're going to assume that this DNA sequence is the template being used to build a messenger RNA. And so what's the corresponding messenger RNA sequence? Well, you remember the G, the guanine, is going to correspond to a cytosine. Adenine, the complementary one. The adenine is going to be complementary to not thymine. No thymines in there. Is the uracil. You got to keep that in mind. That's an easy mistake to make. Thymine is in DNA only. Uracil is in RNA only. So this first one's going to be CUU. And then the CCC, well, that's going to be GGG. And then TTT, well, that's going to be AAA, the adenine. Okay, so it's all, each step is going to be the complementary one. What are the anticodons for the transfer RNAs? Well, the anticodon is going to be similar to the DNA sequence. All right, so the GAA is going to be the GAA is going to be the anticodon for CUU, okay, and CCC, which is up here, is going to be the anticodon for the UU. Well, we said the GGG, and then the AAA is going to be, or the AAA is in the messenger RNA sequence, so the anticodon is going to be UUU. It's going to be the uracil. And what's the amino acid order? And well, we can let's just go on to the next slide, where you see what I'm doing here. The anticodons, we could well, we don't really want to put these in order. We don't want to put these in a sequence like this because the transfer RNA, it's just the anticodon. That's it. So there's 3 anticodons that are not linked together. They are the codons. These triplets are linked in the DNA and the RNA. And if you look at the CUU, which are the codons, you're going to find leucine for the CUU. You're going to find glycine for the GGG and lysine for the AAA. So that's what you're going to, the amino acid order. There are some antibiotics that inhibit protein synthesis, and here's a few of them. And what they do, these are going to target bacterial cells, but these are some antibiotics, and you've probably heard of most of these, maybe all. Chloramphenicol, which inhibits peptide bond formation, prevents binding of transfer RNA, and erythromycin prevents translocation of the ribosome. Puromycin causes causes an early release of the incomplete protein. Streptomycin prevents the proper attachment of the initial transfer RNA, which would be the start codon. And tetracycline, which prevents binding of transfer RNA. These are the mechanisms they work by. A lot of medications are that way. They will interfere with a specific, they will either interfere with or accelerate some biochemical reaction, some biochemical process. And in the case of antibiotics, you want these things specific to bacterial cells. Its antibiotics are, well, they're not necessarily easy to develop, and you've probably heard of the antibiotic resistance to antibiotics. You know, a lot of bacteria have developed ways around these things, but the actual synthesis of the protein synthesis in bacteria and in eukaryotic cells. Eukaryotic means a true nucleus. Okay, they have the nucleus. We're eukaryotes. We have eukaryotic cells. The sequence that we described in this chapter, in this section, is pretty similar, but it is not identical. So bacterial cells have a very similar process. The steps are a lot alike, but there's differences which can be exploited in using they can be exploited in antibiotic synthesis. It's not quite as easy when you're talking about protozoa, fungi, things like that, but that's one of the things that they wanted you to know here. Okay, so we're getting near the end of this, and they want to, they're asking you here, place the following statements in order of their occurrence in protein synthesis. Okay, well, you see there, it's always going to start with DNA, at least the way we're doing it. At least we're in the human body. We're going to, there's a little wrinkle when we get into retroviruses, which you're going to see eventually here. DNA is going to produce our messenger RNA, so that's going to be number one. And then the messenger RNA is going to, they don't say anything on this list for the messenger RNA leaving the nucleus. So the next step that we see here for sure is that the messenger RNA attaches to a ribosome. Once the ribosome is attached, you're going to have a transfer RNA. It's going to bring an amino acid to its codon on the messenger RNA, starting with methionine. This is going to be a start codon. And ribosome is going to move along, and the completed polypeptide chain is going to be released after it hits a stop codon. All right, those are and you can, this is easy enough to reason out. If it is, go through it a few times. These diagrams are pretty complicated because these are big molecules, and drawing them in a kind of a representative form can be kind of complicated. But the basic process, no matter how large the protein is going to be pretty much the same. You're going to get the DNA, you're going to have messenger RNA attached to the ribosome, and you're going to start adding amino acids, hit a stop codon, gone. Okay, and I guess I didn't, in that statement, I didn't mention the transfer RNA, but that's what's happening. Once the messenger RNA is in the ribosome, the transfer RNAs start coming in, start getting attached, and go through this process. Okay, so you see those are what the sequence we had. Okay, this was actually this might have been one of the longer ones that we had. The next section is going to be 17.6 genetic mutations, and this is going to be a little bit involved, but you already know enough to understand what's going to be going on. A mutation is essentially a change in the nucleic acid sequence, and they can have good effects, they can have no effects, and they can have bad effects. So we'll talk about that when we get to that section, which is going to be the next video.